My name is Dr. Matt Rasband. I'm a professor of neuroscience at Baylor College of Medicine. I'd like to invite you, as a prospective student, to learn more about the efforts that we're making here at Baylor College of Medicine to develop new technologies to push the boundaries of modern neuroscience, from my colleague, Dr. Francois Saint-Pierre.
I've had many exciting collaborations here. That's why I came to Baylor. One of them is to build and characterize sensors that are called genetically encoded voltage indicators. That's a mouthful, but it basically means like little proteins that blink when neurons are active. And that permits us to figure out like which neuron is active and where and how they talk to each other and how they process information. So my lab has been spearheading the development of these. So we put little mini proteins together to form a sensor, a bit like Lego bricks. And then we work with collaborators here at Baylor and also outside to test them in model systems, such as like the mouse. Another collaboration, completely different, is with a lab at Rice University, the Egoshen lab. So we've been developing genetic circuits. So they're basically pieces of DNA that allow us to control the way a certain protein is being expressed. So for example, when we express voltage indicators I discussed, or other tools like optogenetics, it's important for all these tools to be expressed just at the right level. So you have enough that you can see your signal, but not too much that it kills the cell or it affects their physiology. So we've developed some tools that can like fine-tune the expression of these various proteins in individual neurons. They've been helping us with the computation, so the modeling, and we've been putting it together, splicing, putting together the pieces of DNA and actually carry on the experiments to test them. So there's been a lot of new exciting development in neuroscience here, I think at Baylor and in my lab. So some of them we've discussed already in the collaborations. One is, so for example, to genetically encoded voltage indicators I discussed, they are, say, like these proteins that blink in response to neural activity. And so before that, one of the key ways you could look at voltage, like direct neural electrical activity, which is the way neurons talk and process information, was using electrodes. So you would put these electrodes in the brain, but as you can expect, they're really invasive. And also, it turns out like in the brain, we don't just have one kind of neuron, one kind of brain cells. There are many, many kinds. With an electrode, you don't know which type of neuron you're listening to. It's just whichever one is close to the electrode. So with these voltage indicators we developed, because they're made by the cell itself, we can use some genetic trickery or wizardry to have them only being produced by the cells that you care about. So now this is helping researchers to listen in and record what goes on in specific populations of neurons to figure out what they do, what's different between them, what's their respective functions. So another example of a new technology that we've recently reported in Science Advances is a new screening technique. So one of the limitations when we're building tools or when we're doing discovery-based research, how quickly you can get the results. So, and in particular, what a lot of researchers want is to map what the genotype, like the DNA sequence, to the function, what's called the phenotype. Before that, there was not a lot of tools that allow us to do this. And so we've developed a new process called Spotlight, which allows us to image cells that it can be in vitro, and we're also extending it to in vivo in animals. You can image cells doing interesting things. And if you want to figure out what's different about their genetics or their proteome, like what's different about these cells, you can zap them with light, and then we can recover them specifically and then sequence them to figure out what was different about them. Technology is really key for neuroscience, and as you might know, like the brain hasn't evolved to make it easy for us to understand, right? Like there's no interface with researchers where you can easily look at what's going on and also perturb, make some changes to see what types of neurons or what areas do what. And it's also a big blob that's very opaque, it's not transparent. So there's definitely still a lot of needs to being able to interface with the brain in order to figure out how it works in both health and in disease. A lot of the investigations so far have have focused on very superficial layers of the brain, because if you go deeper, then if you want to look at deeper layers or nuclei or the brainstem and so forth, you typically have to dig in. You have to put some electrodes or you have to put some fibers, but you have to make some damage in the overlying tissue, which is not ideal. We don't want to disturb the system that we're investigating. So some of the future is about the developing tools that are going to allow us to pierce and image or perturb deeper in the brain. Another is going for longer, say like developing tools that allows us that work for extended duration, so they don't become dim, they don't break over the course of an experiment, so going longer. And also going faster. So a lot of what goes on in the brain is fast. It's millisecond time scale.
I hope that you've enjoyed this overview of our neuroscience graduate program here at Baylor College of Medicine, and that we've convinced you that this is an amazing place to train in the neurosciences. Lastly, I hope that I'll see your application.